Assalamu alaikum. So let's go into something really exciting. So let's talk about DNA cloning. Okay, let's define DNA cloning. So DNA cloning is basically a technique that we use to make pieces of many copies of a piece of DNA. And we use a certain biological system for that. Now, we can use bacteria mainly. We can use yeast as well because they're easy to manipulate, they are easy to handle. Or we can do DNA cloning on human cells in culture, in laboratories, that is. So, or we can use a cloned DNA to have bacterial cells or human cells express specific genes, like the insulin gene, for example, or a gene that produces an enzyme or whatever. So, and this is done by creating a recombinant DNA, which is basically a DNA that is a that is a that is made of different pieces of DNA from different sources. So we say human DNA, and we combine it with bacterial DNA. So that's a recombinant DNA. So basically, we have a piece of DNA that contains another piece of DNA from a different source. Now, we have one DNA that is called a vector. It's the carrier. And we have the gene of interest, or the piece of DNA of interest. Now, usually it can be a gene that produces a protein, or it can be non-coding RNA, okay, that is not translated, whatever. And in order to do this, we use restriction endonucleases. So let's start talking about, let me remind you of restriction endonucleases and what they mean exactly. So restriction endonucleases are bacterial enzymes that cleave DNA. And what they do, basically, well, they recognize restriction sites, short DNA sequences, and they cleave within these sites, forming restriction fragments. Okay, and these are natural enzymes that exist in bacteria. And the natural purpose of these enzymes is to cleave bacteriophage DNA. So bacteriophages are viruses that can infect bacterial cells, and they take over these bacterial cells. They infect these bacterial cells. So they insert their own DNA, and then they take over. So what bacterial cells do is that they express restriction endonucleases that cleave the bacteriophage DNA so that they cannot control these bacterial cells. So humans identified these enzymes, and they said, how can I take advantage of these enzymes? So they use them for cloning. Now, what I did not tell you is the following. Actually, I told you that before. Basically, these restriction endonucleases can cleave restriction sites that are palindromic, meaning that, well, they have palindromic sequences, meaning that you can read the restriction sites from either DNA strand the same exact way. So here we have GAATTC, and you read it the same exact way from the other complementary strand, GAATTC. Now this is the restriction site of ACAR1. Hindi 3, small 1, also recognize palindromic sequences as well. Okay, this is what I did not tell you before. And that is, these restriction endonucleases can make blunt-ended restriction fragments or cohesive-ended DNA fragments, restriction fragments. What we mean by that is, with the blunt ends, is that the cut occurs right in the middle, in the center of the restriction site. So EcoR5 can recognize this sequence, GATATC. Now notice again, it's palindromic, right? GATATC. the cut occurs between T and A, right in the middle, okay? And note the DNA that the restriction fragments have blunt ends, that is, the cuts are straight, right in the center, okay? On the other hand, note here, echo R1 makes a staggered cut, meaning that the cut occurs not in the center, rather on the sides. So the cut occurs between the G and the A. The G and the A, note that are not in the center, not in the middle of the restriction site, rather on the sides, creating cohesive ends. Ends that have overhangs, right? Now they are called cohesive ends because these ends are complementary to each other. So you have TTAA, and here you have TTAA. So the AA here can hydrogen bond with the TT right here. The TT here can hydrogen bond with the AA right there. So these are known as cohesive ends. Okay, now I'll tell you later on that these hydrogen bonds are reversible, right? So the two pieces of DNA can separate from each other because the bonding is reversible. Now, in order to have the two strands connected to each other again, you need the formation of phosphodiester bonds between this fragment and that fragment, between the A and the G, the A here and the G there. Now, if we have two different pieces of DNA from different sources, and we have them cut by echo R1, we're going to have the formation of several restriction fragments, but all of them are going to have cohesive ends. So we can have the formation of hydrogen bonds between these two different fragments from different sources because they have ends that are complementary to each other. And then we can form, we can have the formation of phosphodiester bond between the A and the G, right here. So we have what is known as a recombinant DNA. And a recombinant DNA is a DNA that is made from different sources. And we use restriction endonucleases to create these DNA molecules. So note that I have now one piece of DNA that is generated from different DNA sources. Now again, we can have the addition of a DNA ligase in the presence of ATP because it's ATP dependent, so that we have the linkage, the permanent, strong linkage of this phosphodiester bonds between the DNA fragments. Okay, so now we have what is known as DNA ligation. The enzyme is known as DNA ligase because it ligates, it connects molecules with each other. So what is DNA cloning? Well, DNA cloning means that you make copies of one thing. It's like a copy machine, okay? You have this piece of paper and you make 10 copies, 100 copies of that same piece of the paper. So what we do is that we can use bacteria. So these bacterial cells, okay, eventually you can grow them on dishes, and we have this bacterial cell forming a clone. Okay, so this is the bacterial clones, or the different bacterial clones. Each clone comes from one single cell. So these all are identical to each other, and identical to the original cell. Now, these cells right here are identical cells because they can come from one bacterial cell, so they are cloned. Okay, so if I say human cloning, it means that I'm making a copy of a person. So I have the same person copied over and over again. We have many copies of that same person, meaning that this person has identical DNA, identical genetic material. Before talking about how we clone DNA, we have to talk about DNA plasmids. These DNA plasmids are vectors, okay, vectors, meaning that they are carriers of the DNA that we want to clone, the DNA piece that we want to make um, many copies of. Now, these DNA plasmids can be cloning vectors or expression vectors. Okay, we'll talk about the difference between each. We'll talk about cloning vector. We'll talk about using plasmids as cloning vectors. Okay, so the idea here is what a plasmid is. It's extra chromosomal DNA in bacterial extra chromosomal DNA, meaning that if you look at a bacterial cell, we notice that each one of them has its own chromosome, the main genetic material that contains like E. coli, containing 4,000 different genes. So there's one copy of bacterial chromosome, or chromosomal DNA. But bacterial cells can also have plasmids. These are circular DNA pieces that can replicate independently. So each bacterial cell can have multiple copies of each plasmid, and these are small pieces of DNA. Now, naturally, plasmids contain genes. They carry genes that can give advantage to bacteria. For example, it can make them resistant to viruses. It can make them having antibiotic resistance ability. Okay, so that's what's important about is that they can replicate independently of the chromosomal DNA, and they can be engineered as well. Okay, that is, we can modify them, which so that they can do certain things. Okay, and when we say modify them, this is exactly what we're talking about. We're talking about genetic engineering. That is, we engineer genes um, or pieces of DNA, such as plasmids. Okay, so that's what plasmids are. Now, what we do is that we take these plasmid molecules and we make them function as vectors, meaning that we make them as carriers of pieces of DNA. Target genes, for example, whatever piece of DNA that we want them to carry. And we create recombinant DNA, DNA made of different sources. So we take these plasmids, which are circular again, and we open them up with a restriction enzyme. We also take the target DNA and we add the same restriction endonuclease. So now both the plasmid DNA and the fragment DNA that we're interested in cloning have cohesive ends, and these cohesive ends are complementary to each other. Okay, so we open them up with a restriction endonuclease, we combine them with each other, we just put them together in the same tube, and we add a DNA ligase. And what happens is that these ends, cohesive ends, are complementary to each other, so they can combine, they can hydrogen bond with each other. When we add a DNA ligase, the DNA ligase would form phosphodiester bonds between the two DNA pieces, and here we have a recombinant DNA, a plasmid DNA. That is, the piece of DNA that we're interested in now is permanently part of the plasmid. That is what we mean with recombinant DNA technology. Recombinant DNA technology, the creation of recombinant DNA, the creation of DNA pieces that are made that come from different sources. So, in order to utilize plasmids as vectors, they have to have at least three features. One, they should have their own origin of replication. right? Remember that origin of replication, that is the starting point of DNA replication. So go back to the DNA replication lecture and read about the origin of replication. So they have their own origin of replication, at least one. All they need is just one. They don't need more than one, just one. And this is the starting point of DNA replication, and that what makes them replicate independently of the chromosomal DNA. They should also have a cloning site. Okay, that is a site where that is recognized by it's a restriction site, a site that is recognized by a restriction endonuclease. Now the site is opened by this restriction endonuclease, creating cohesive ends, and so that when we add the DNA that we're interested in cloning, they can hydrogen bond with each other. And the third thing is that plasmids should have antibiotic resistance gene like ampicillin. Ampicillin is an antibiotic, and this gene makes bacteria resistant to ampicillin. Okay, that's exactly what we're talking about here. So that only bacteria that have this plasmid can survive ampicillin. They can, if we add ampicillin to this bacteria, they can survive. They cannot be killed. So these are the three things that are needed by plasmid DNA. A, origin of replication, so that they can replicate independently of the rest of bacteria. A cloning site, restriction site that can be cleaved by restriction endonucleases, so we can insert the DNA piece of interest, and the antibiotic resistance gene, so that only bacterial cells that contain the plasmid would survive if we add an antibiotic to them. So that's basically what we do. Okay, so we take the plasmid DNA, we take our insert, we add ECAR1 to each one of them, creating cohesive ends. Now these two ends are complementary to each other. When we add them to each other, they're gonna hydrogen bond. We add a DNA ligase, and the DNA ligase forms permanent linkage between the plasmid and the DNA of interest. Again, here we go. So we take DNA from human cells. We add ECAR1, let's say. Okay, again, we can add any restriction endonuclease. Cohesive ends are created. We add the same endonuclease to the plasmid, creating cohesive ends as well. When we combine them to each other, okay, oh, sorry. So here we take the plasmid from bacteria, and we cut the plasmid with ECAR1. We combine them with each other, in addition to a ligase, and here we have the recombinant DNA. The recombinant DNA is then what we call transformed into bacteria. Transformed into bacteria. That is, we insert the plasmid into bacteria, and there are different ways of doing that. Now, the bacterial cell has a plasmid, has a recombinant DNA. Okay, now, again, same idea. Now, let's say we have bacteria cells that have a plasmid, and we have bacterial cells that do not have a plasmid. Now we add ampicillin to these bacteria. Now this bacterial cell would die because they are not resistant to ampicillin, and this bacterial cell would survive. Okay, because they have the plasmid that contains the recombinant DNA, the ampicillin resistance gene. Now we can multiply these bacteria, we can grow these bacteria, and let's say that we do the inoculation at 4 p.m. 4 in the afternoon. We let bacterial cells grow overnight. We're going to have millions of bacterial cells after like 16-18 hours. Now each bacterial cell would contain at least one plasmid, and these are the ones that would survive ampicillin. Now we're going to have billions of plasmid molecules, recombinant DNA molecules this way. So here we have a recombinant DNA, and we can clone our DNA of interest. Okay, so let's talk about these DNA inserts. What do we mean by DNA inserts now? We'll talk about libraries. Now, DNA libraries are basically, well, it's the same concept as a library, where, for example, you're interested in reading a book, so you check where this book is, and you find out that the book exists on the fourth floor, rack number whatever, shelf number whatever. So you go there, you pick up the book that you're interested in. And the same idea exists in DNA. That is, we have plasmids that can contain, each one of them containing a different a DNA piece that is different from other plasmids. So, and we can uh, uh, store these plasmids in fridges, on shelves, whatever, you name it. And the idea is that if I'm interested in studying a certain piece of DNA or a specific gene, we can go to room number whatever, shelf number whatever, and we take the tube that contains this plasmid of interest. So, and the idea, and there are two different types of libraries. There are genomic libraries and there are cDNA libraries. Now, let's start with genomic DNA. Genomic DNA is basically, well, the whole DNA of a cell or a group of cells. We take this genomic DNA and it's cut by a certain restriction endonuclease. We have a plasmid, we combine these cut DNA molecules with each other. Now we have each DNA piece inserted into a plasmid. We take these plasmids and we transform bacteria. And each bacterial cell would contain a recombinant DNA, a plasmid DNA, that is different from the other cell. Now, each one of them would have a different piece cloned into the plasmids that they carry. So that's genomic DNA library. Now, I would know later on what each bacterial cell or clone contains exactly, what piece of DNA they contain. Okay, and anytime I want to study this piece of DNA, all I need to do is take this bacterial cell or cells and grow them up, and I'll have millions and billions of copies of the plasmid DNA that contains the piece of DNA that I'm interested in. Well, let's talk about the cDNA libraries then. cDNA libraries are created from messenger RNA. So here we're talking about expressed genes. Okay, so we take the messenger RNA from cells, we convert them to complementary DNA molecules. These are called cDNA molecules. Complementary DNA because they are complementary to the messenger RNA molecules. So we convert mRNAs to complementary DNA molecules. And we use a special enzyme known as reverse transcriptase. Reverse transcriptase. So a reverse transcriptase, is a viral enzyme. So note how we take advantage of creatures, God's creatures. So we take this viral reverse transcriptase, and this is the enzyme that converts messenger RNA into cDNA. It can convert actually any RNA molecule into DNA. So it's the reverse of transcription. Okay, and that's why we call it reverse transcriptase. So we convert messenger RNA to cDNA. Okay, and we have different genes, right? We have different messenger RNA molecules. We have different DNA molecules now, complementary DNA molecules. We add the restriction endonuclease, creating cohesive ends. Same thing with plasmids. We combine them with each other with DNA ligase, and now we have plasmid molecules, recombinant plasmid molecules. Each one of them carries a gene. Okay. And we can transform bacterial cells with these plasmids. Now each bacterial cell now contains recombinant plasmid DNA that carries a specific gene. Okay, so we call this cDNA library. Now I know what each bacterial cell or clone carries, and every time I want to study a gene, all I need to do is pick up this bacterial cell to grow them up. Now I have millions, billions of copies of the plasmid DNA molecules of interest. Again, genomic DNA. We take genomic library. Where genomic DNA is cut by a restriction endonuclease. The same endonuclease is used to cut plasmid DNA. We combine them with each other. Now we have different DNA molecules carrying different fragments of genomic DNA of different sizes, of course. Okay, and we transform these bacterial cells with the plasmids, and each clone carries a piece of genomic DNA that is different from the other cells. Now with the cDNA library, we take the messenger RNA, and this messenger RNA is converted into complementary DNA or cDNA, and we do the same thing. We cut the DNA pieces with the restriction nuclease. We have recombinant DNA. Each piece of plasmid molecule contains a specific gene. We transform bacterial cells again, and we have cDNA library. Okay, again, you can read this on your own. It's the same basic idea, but it's written and different figure. Same idea. That's creating a cDNA library. Again, same idea. Okay, so you can read it on your own. Now, let's talk about this point, and this point is really important. What are the differences between cDNA library and genomic library? Now, notice that with the genomic DNA, we take everything. Every DNA, we take DNA, genomic DNA. We take everything. So we can have pieces that can represent genes, exons, introns, centromeres, telomeres, promoter proximal elements, enhancers, everything. Okay. Now, with the cDNA library, we're only taking the expressed genes. So if I take from a person a nerve DNA, genomic DNA, from nerve cells or from epithelial cells, skin cells, or whatever, and I cut the genomic DNA with a restriction in the nucleus, and I use it to create recombinant DNA or to create a library. Now, these libraries are going to be exactly identical or the same because they all contain the same DNA. But if I create a cDNA library from nerve cells, it's going to be quite different from a cDNA library created from skin cells because these different cells express different genes. Again, we have 20,000, let's say 20,000 protein coding genes, right? So we have 5,000 genes expressed in nerve cells. We have 6,000 genes expressed in skin cells. They're not going to be identical. Some of them are going to be the same, like histone genes, for example. Actin genes may be identical, and so on. But there are specific genes that are expressed in skin cells, and these are different from genes expressed in nerve cells. Okay, like neurotransmitters, for example, like the enzymes that create pigments in skin cells, and so on, right? So basically, cDNA libraries are going to be different when created from different cells. Now, the other thing is cDNA libraries would contain things like exons only. They're not going to have promoters because they are not expressed. We're talking about RNA. representing messenger RNA molecules now. Okay, we're not talking about any type of RNA. So here we're talking about protein coding genes starting with their messenger RNA molecules. So they're going to be, the cDNA library is going to represent exons. There will be no promoters, there will be no enhancers, no promoter proximal elements, no centromeres, no telomeres, just exons, no introns even. Okay, so keep that in mind. Right. So let's talk about something different here. Let's talk about how we can analyze transcriptional regulatory sequences. Now, we talked about DNA, regulatory pieces of DNA, like promoter regions, for example, or enhancers. These are binding sites for specific promoters, for specific regulatory proteins. General transcription factors, specific transcription factors, like cyclic AMP response element binding protein, for example. It binds to a specific promoter proximal element. There are specific proteins that bind to enhancers. Okay, these proteins cannot bind to other enhancers. Well, so let's say that I want to study how good a gene is expressed in a certain cell type at certain conditions. I don't know, in the presence of hormone versus absence of this hormone, let's say. In the presence of light versus in the dark, and so on. So we want to study how good, how efficient a promoter is at certain conditions in expressing a certain genes. Now, note, I am not interested in studying the gene itself. I'm interested in studying the promoter of that gene, not the gene itself. Okay, but I want to study how good a promoter is at certain conditions. So we can use DNA cloning, the creation of recombinant DNA, to do so. So what are the different transcriptional regulatory sequences? Well, here we're talking about the promoter, we're talking about the promoter proximal elements, we're talking about enhancers and silencers. So we have a gene, we have transcription starting point right here, right? And right before the transcription start site, we have the different regulatory sequences. You have the promoter, promoter proximal elements, enhancers, and so on. Now we want to study this, let's say we want to study this region right here. Now note that we can have silencers instead of enhancers, okay? So let's talk about something different now and see how we can combine all this knowledge together. So we take advantage of an enzyme that is expressed in fruit flies. Okay, so fruit flies are these insects and their butts glow, right? They light up, creating this beautiful scene at night. And the enzyme is called Luciferase. Luciferase, okay, and what it does is that there is a substrate called luciferin, and this luciferin is cleaved or converted by the luciferase enzyme into oxyluciferin, and this is the molecule that lights up. So I don't care about the reaction itself. All I care about is that we have this enzyme and it cleaves luciferin, creating a product that lights up. Okay, so how can we take advantage of all these tools, molecular tools? So that's what we do. Okay, we create recombinant DNA using these plasmids. So we have a plasmid, bacterial plasmid, and we take the fruit fly luciferase gene and we add a promoter that we want to study, So we're not using the promoter, the original promoter of the luciferase gene, right? So we have it under control of a promoter of interest. So let's say that we have the luciferase gene be under control of the enzyme, the hydroxylase enzyme, the dopamine hydroxylase enzyme, for example, the promoter of the dopamine hydroxylase gene. Okay, and so we create this recombinant DNA, and we transfect, we insert the plasmid into a human cell, a nerve cell, let's say. Okay, so now this nerve cell has its own DNA of interest. Okay, now we can place these cells under different conditions. Okay, and we can then study the expression of the luciferase gene, which is under the control of the promoter of interest, under these conditions. In the presence of light versus dark, in the presence of a hormone, in the presence of a certain neurotransmitter, stress, you name it, okay, you name it, it doesn't matter. Now, we can then add luciferin, and luciferin, if luciferase is expressed, it's going to convert luciferin to this molecule that lights up, the product that lights up. Now, the stronger the signal, the stronger the light, the more the gene is expressed, or let me say, the more the gene is, the higher the activity of the promoter is, the more the gene is expressed. The more luciferase is in the cell, the more light is gonna be produced. Okay, so that's the idea. So we place these cells in different conditions. In the presence of hormone versus absence, neurotransmitter, yes, no, stress, yes, no, and so on. And we measure the amount of light. And that gives us a signal, an idea of how this promoter is regulated at different conditions. Okay, that's one thing. Something else is that we want to study this regulatory sequence. Totally different idea now, okay? We want to study what we know that this region contains regulatory sequences. Promoter, proximal element, promoter, enhancer, whatever. I want to study the position, the location of all of these regulatory sequences. The exact position of the core promoter, the exact position of an enhancer, the exact nucleotides that control each one of these. So we do this. So we have now the luciferase gene, which is right here, the luciferase gene under control of a promoter of interest. And I don't know what the different sequences represent. Now I want to say, here we have a repressor region, here we have an activating region, and here we have the core promoter sequence. Now I don't know what the positions of these sequences are, and that's what I want to study. So I take the DNA piece that contains what I think is the promoter region containing all of these regulatory sequences. So I take it, I clone it into a plasmid. I clone it into the plasmid that contains the luciferase gene. I create a recombinant DNA here. And I take this recombinant DNA this plasmid that contains the luciferase gene that is under control of the promoter that I want to study. Okay, and I inserted or transfected into certain cells, nerve cells, skin cells, stomach cells, whatever, and I study how the luciferase gene is controlled by these sequences. And what I do is the following. So I take the luciferase gene, the recombinant DNA that contains the luciferase gene, and I place the promoter that I want to study. Okay, the complete promoter. Okay, this whole thing that contains the core promoter, the activating region, the repressing region or repressor region. And I start creating different fragments of this right here. So I start chopping up, I start deleting different regions. So for example, I remove the part of this region, I remove this region, I remove this whole thing, I remove this thing right here, I remove this core promoter complex, and so on. So I have, again, the luciferase gene under control of the whole promoter region, the whole thing right here. And then I start chopping up this piece of DNA. So I remove this part right here. Then I remove this part, creating this. I remove this part right here. So I have now the luciferase gene under control of the core promoter and the activating region. And I remove this part, I remove that part, I remove this whole thing, and so on. Now here I have a piece of DNA that contains no promoter whatsoever, just that luciferase gene. Now the luciferase gene is right here. So this piece of DNA right here is represented by the black arrow. Okay, now, so let's look at the results. Now I transfect, so I have different plasmids, different promoter, pieces of promoter, the same luciferase gene, nothing is changed with the luciferase gene, and I transfect these different plasmids into cells. So each group of cells would have a recombinant plasmid DNA containing the luciferase gene and different pieces of the promoter. Okay, and I measure the amount of light that is produced by by converting the luciferin substrate into the light product. Okay, great. Now, I have to have controls. I have to have things that either work or do not work, so that I can compare my experimental DNA with the controls to make sure that the conditions of my experiment are fine. So I create negative controls and I create positive controls. That is, something negative control is something that I know does not work. A positive control is something I know works for good. Now, if the negative control works and it produces luciferase, it means that there's something wrong. If the positive control that should work, if it doesn't work, it means that again there is something wrong with the DNA that I've prepared. Okay, so here we have the luciferase gene with no promoter whatsoever. Oh, I know that the luciferase gene does not have a promoter. That's a negative control, something that I know does not work. And here we have the luciferase gene that is under control of a good promoter, a different promoter, a promoter that I know works great. So that's a positive control, just to make sure that the conditions of my experiment are fine. So, and that's the amount of light right here. Okay, so here we have the luciferase gene under control of no promoter. There is very little signal. So someone would say, whoa, wait a second. There is no promoter. Why is there a signal? Well, remember when I talked about leakage? of promoter regions, genes can be expressed even though they have no promoter, even though there are no activators, and so on. Well, there's little signal. So that's a negative control. That's what no expression means. Well, there is some expression, but that's what no expression means, because now the luciferase gene is not under control of any promoter. Okay, here we have luciferase gene under a good promoter, different promoter. Again, that's a positive control. There is excellent signal right here. Again, there's a lot of expression, 100%. So that's my 100%. Okay, now here we have the luciferase gene under control of the promoter that I'm testing under certain conditions, the whole promoter. Okay, here we have a signal. That's what an expressed luciferase indicates when under control of this promoter under study. Okay, so we have this signal right here. Okay, then what I start to do is I start removing pieces of DNA, pieces of the promoter region. So I start removing the repressor region. So what do you think would happen? If I remove the repressor region, I should see an increase in expression, because now there are no repressors that can bind to the promoter and can inhibit the expression of the luciferase gene. Okay, so by removing the repressor region, I should get increase in expression, and that's exactly what happens. So when under control of the whole promoter, including the repressor region, I get this amount of expression. If I remove the repressor region piece by piece, little by little, I get more expression. Wonderful. So I know that this region contains a repressing region. Okay, now let's say that I start removing, now I remove this whole thing, right? This is what I get. Now there's basically, by the way, no difference between this and that. Okay, so this is just experimental conditions. Now I start removing the activating region. What do you think would happen if I remove the activating region? I should see a reduction in expression, and that's exactly what I see. If I remove the activating region, there is reduction in expression. There of luciferase again. Okay, there is reduction in the amount of light that is produced. Okay, so and that's exactly what we see. There is complete reduction in expression of the luciferase gene by removing the activating region. So I know that this region contains an activating region, because when we remove it, there is no expression of the luciferase gene. What would happen if I remove the core promoter? Even more reduction of expression of luciferase, more production, less production of light, and that's exactly what I see. This here is similar to this right here, right? Because that's what no production of luciferase means. Very little production of light. Same thing here. Okay, so by removing the core promoter, there is no expression whatsoever. So this is called the luciferase reporter gene assay. The luciferase reporter gene assay. Assay means experiment that measures something. Reporter gene is a gene that gives us an idea. It reports something. And this gene is the luciferase gene, the fruit fly gene, that converts luciferin into a product that produces light. The more luciferase we have, the more light we have. So here we have an experiment where we want to identify the function of different promoter regions, different pieces of a whole promoter, whether this sequence is whether the sequence represses, inhibits protein expression, or activates protein expression, or if it is essential for gene expression, um, that is the core promoter. Or we can place it inside cells, we can transfect cells, and we can put these cells under different conditions again, in the presence of hormone or no, stress or no, nutrition or no, you name it. Okay, and we can measure how a promoter, a specific promoter, functions, how it behaves under different conditions by measuring the amount of luciferase that is produced, which is represented by the amount of light that is produced as well. Allah